This is the third video on the March 2024 paper 4-2. Now, describe means no biology. I keep on telling this to you again and again, and it's one of the ones in which you can score full marks. All decrease in absorbance with time. The general trend. All decrease in absorbance with time, all decrease at a constant rate. Then green has the highest absorbance throughout, and absorbance stays fairly constant, and small rate of decrease in the green. Then you give me a data quote of two colors. Like for instance, green was in 4 minutes 1.44 and blue was 0.90 and red was 0.70. So data quote is that means you give me two colors compared to support marking point 1 or marking point 3. Marking point 1 was all decrease in absorbance. Marking point 3 was green light is the highest absorbance. So at 0 it was 1.5, and at 4 minutes it just decreased a little, 1.44. So it was had the highest absorbance. Blue light, red light, and then you can see the graph how the absorbance is different for all the three different wavelengths of light. So all decrease in absorbance at constant rate, green highest absorbance, smallest rate of decrease in green 1.44, blue 0.90, 0.70 at 4 minutes. Then coming to part 3, with reference to the light dependent stage of photosynthesis, explain the differences between the results shown in figure 7.1 for red light and green light. There was, you see, light dependent state, what happens? More light energy absorbed by chlorophyll, more photoactivation or more electrons emitted. So more rate of electrons transferred to DCPIP. So more decolorization of DCPIP. So more light absorbed by chlorophyll, more electrons emitted, more electrons transferred to DCPIP, and a faster decolorization of DCPIP. Now, coming to the B part of the question, changes in the atmospheric carbon dioxide concentration, light intensity, and temperature can affect the rate of photosynthesis. These three factors directly affect different processes of photosynthesis. Complete table 7.1 using a tick to identify the process that can be directly affected by each factor, or a cross to identify the processes that are not directly affected by each factor. Now, this is written in bold, so we need to read this carefully. Indirect effects, where a change in the rate of one process affects the rate of a different process, should not be considered. A tick or a cross must be placed in the final column of every row. Now, carbon dioxide concentration, does it affect the Calvin cycle? Yes, it does. Does it affect photophosphorylation? No, it does not. Light intensity, does it affect the Calvin cycle? No. Indirectly it does, because you see, light intensity, more the light intensity, more ATP and NADPH, so more Calvin cycle. But it said in the question, it does not, should not be considered if it is change affects the rate of a different process. Indirect effects will not be. Then light intensity, does it affect photophosphorylation? Yes, of course. More the light, more the photophosphorylation, non-cyclic and cyclic photophosphorylation. Then temperature, does it affect the Calvin cycle? Yes, it does. Photophosphorylation, yes, temperature affects that, because there's an enzyme required in photolysis of water. And also ATP synthase, and all these are required for the photophosphorylation. So we have a tick. Now this was for 3 marks. So that means this is 1 mark, this is the second mark, and then this is the third mark. So you have to understand that this must be a very easy level, so that's why there's less marks for it. Question number 8. Approximately 2 into 10 raised to the power 9 people in the world are currently infected with the bacterial disease tuberculosis caused by Mycobacterium tuberculosis. Early diagnosis is important so that treatment can begin. APOPO is a non-profit organization that has trained African giant pouched rats, Chrysitomus gambianus, to use their sense of smell to detect M. tuberculosis. They do this by sniffing a sample of thick mucus from the lungs of people who may have TB. The African giant pouched rats are able to detect the presence of Mycobacterium tuberculosis with an accuracy of 87 to 93%. Figure 8.1 shows an African giant pouched rat. The type of receptor used by the African giant pouched rats to detect Mycobacterium tuberculosis is the same as that used in human taste buds. Name this type of receptor cell. Chemoreceptor. It is used in taste buds, like, so it's a chemoreceptor. Like you put salt on somebody's tongue and you just blindfold them and say, okay, what is it? And it'll say it's salty, or it'll say it's, if you put something sugary, it'll say sugar. So that's a chemoreceptor. It is actually chemicals, detects chemicals. Suggest why African giant pouched rats trained to detect mycotuberculosis may also be able to detect other species of mycobacterium that cause TB. The reason would be that different mycobacterial species produce the same chemicals or the same proteins. Since they share common ancestors or the same genus, they share many genes, and same chemoreceptors will be stimulated. So produce same chemicals slash proteins, share many genes, are closely related, the same chemoreceptors will be stimulated. The African giant pouched rat belongs to the kingdom Animalia in the domain Eukarya. Complete table 8.1 to show the full classification of the African giant pouched rat. Now that was easy. This would be phylum, class, order. And there's a mnemonic that you need to remember: family, genus, and then the genus would be what? They have given you the name of it. That will be Chrysitomy. Cricetomy. Now where did that come from? You can see it here. when they talked about it. Here is it. This is the genus, this is the species. Also, you can see that this is for 2 marks. So how am I going to mark it if I'm the examiner? If 4 correct, 2 marks. If 1 or 2 or 3 correct, then it's only 1 mark. Then part C of the question, it says differences between members of the domain eukarya and members of the domain bacteria include the presence or absence of particular membrane-bound cell structures. Outline other differences in the characteristic of features of members of the domain eukarya and the members of the domain bacteria. Well, I would just draw a line and I would write here eukarya. You can actually do that. I don't know why you're all scared to do that. You always ask me that. Where does it say do not draw a line? So what does it have? Linear DNA. And this has plasmids or circular DNA. Or you could have said plasmids present, which are also circular DNA. Then histones, no histones. Then cell walls only in some eukarya, cell walls in all bacteria. Then cellulose in cell walls in certain eukarya. Peptidoglycan in the cell wall of bacteria. Then eukarya divide by mitosis. Bacteria divide by binary fission. Eukarya, some sexual reproduction. Bacteria is all asexual reproduction. Eukarya have 70S and 80S ribosomes. Bacteria only has 70S ribosomes. And eukarya have cells more than 5 micrometers in diameter. They are unicellular, 1 to 5 micrometer in diameter. So a lot of points, and I'm sure you could have got all those points in, and you would have got your 4 marks. So you could do a column like this and say eukaryote bacteria, linear DNA, circular DNA, histone, no histone, cell wall in some, cell wall in all, cellulose, chitin in cell wall, peptidoglycan in cell wall, divide by mitosis, divide by binary fission, 70-80S ribosome, only 70S ribosome, and then of course have cells which are more than 5 micrometer and cells which are 1 to 5 micrometer. I didn't have the space to write that. Then coming to the D part of the question, which is the last part of the question, describe with reference to the structure of viruses how viruses are classified. They either classified the DNA or RNA, and the nucleic acid is either double or single stranded, or you can also say presence in the absence of the phospholipid envelope, or you can the type of disease which they cause, or type of host that they will invade. So any of these points, DNA or RNA, nucleic acid is double or single. You see, here the DNA can also be double or single, and here the RNA can also be double or single. Then type of the disease they cause, type of the host they invade. Question 9. An investigation was carried out to study the effect of different intensities of blue light on the percentage germination of barley seeds. Barley seeds were exposed to blue light for a period of 7 days. All other variables were kept constant. The results are shown. Table 9.1. Light intensity arbitrary 0, dark 98% germination, 36 units 76.9, 48 units 45.0% germination, and 57 14.7. The effect of blue light on the concentration of abscisic acid was also investigated. ABA concentration was measured at intervals over 7 days in barley seeds exposed to blue light at intensity of 57 arbitrary units. The results are shown in table 9.2. Day 0, 100 concentration of ABA, day 1, 90, day 3, 350, then 351, and then 381. For comparison, in the dark, the concentration of ABA and barley seeds fell from 100 AU at the start of day 0 to 45 AU on day 1 and did not increase from day 1 to day 7. So we're getting a whole lot of information which we need to process. So you can see from this information, you can see what is happening. As the light intensity is increasing, the percentage germination is decreasing. Light intensity at arbitrary units is increasing from 0 to 57. Here percentage generation from 98% has gone down to 14.7%. Now Another factor they've given is day 0. You can see the concentration of ABA, that is also increasing. Now, percentage germination is dependent on what? Gibberellins. So somehow ABA is inhibiting gibberellin, because gibberellin is required for, this is what the syllabus, but we've got to study is gibberellin is required for germination. For comparison, then this barley seeds from 100 AU at the day to 1 day did not increase. So the ones in the dark actually did not have any ABA. Here the ABA you can see is increasing. It decreased 1 and then 90 has gone up to 381. Now let's look at the question what they're asking us. It's a ABA is thought to affect gibberellin synthesis or activity. Using the information in table 9.1 and table 9.2, describe the effect of blue light on the germination of barley and suggest an explanation for this effect. So the explanation is important. Describe would be easy, of course. Describe is easy. Blue light decreases germination. As intensity of blue light increases, percentage germination decreases. Then the data quote 0, 98, 57, 40, 14, 57 units, it's 14.7. Now here comes the explanation. As blue light increases the concentration of ABA, ABA inhibits gibberellin synthesis. Now this was given in the question. And then you could have given me any other valid suggestion like DELA not broken down or promotes dormancy or stops gene expression. So any of these points, blue light decreases germination, as intensity of light increases, percentage germination decreases, then the data quote 0 AU 98% germination, 57 AU 14.7% germination, then the explanation light increases concentration of ABA, ABA inhibits gibberellin synthesis, DELA not broken down. Now this is a direct question. The B part of the question, after germination, auxin is important in the growth of barley plants. Describe and explain the role of auxins in cell elongation. Now this is a long question, 7 marks, and there are a whole lot of mark scheme points. There's lots and lots of marks. It's a very direct question. I mean, either you know this or you don't know this. There are 12 mark scheme points. Auxin binds to receptor on the cell surface membrane, stimulates proton pumps. Protons move from cytoplasm to cell wall. pH of the cell wall decreases. Expansins activated. Loosens linkage between cellulose microfibrils by breaking the hydrogen bonds. Potassium channels open. Potassium diffuses into cytoplasm. Water potential of the cytoplasm decreases. Water enters by osmosis. Increase in turgor pressure or increase in the volume of cell. Now here's the mark scheme for this, and I feel you need to memorize some of these points. So the first one is auxin binds to receptor on the cell surface membrane, stimulates proton pumps. Now if you remember, this is the cell wall, and then you have the cell membrane which is inner to it, and the cell membrane of course would have the where the receptor would be present, where the auxin would bind, and then it stimulates the proton pumps and hydrogen ions would be moved into the cell wall. So the hydrogen ions would be in the cell wall, and when in the cell wall, what are they going to do? So stimulates proton pumps, protons move from the cytoplasm into the cell wall, and the pH of the cell wall decreases. So this cell wall, let me make it just a little bit bigger to explain this to you all. So the cell wall will become, pH will decrease, so more hydrogen ions in the cell wall. So pH of the cell wall decreases. Now expansins will be activated. Loosens the linkages between the cellulose microfibrils by breaking the hydrogen bonds. So calcium channels open. Now the calcium channels are separate channels here. Sorry, the potassium channels. Potassium channels open, and potassium ions are going to diffuse in. So potassium ions are going to diffuse in. Cell wall is permeable to everything. The cell membrane isn't. So potassium channels open, potassium diffuse into the cytoplasm. This lowers the water potential. Water potential of the cytoplasm decreases. So then of course what is going to happen? Water is going to enter by osmosis, because we suddenly have a lot of potassium inside the cytoplasm. Now, more solute in the cytoplasm means it will lower the water potential. So water potential of the cytoplasm decreases, water enters by osmosis, increase in turgor pressure or the volume of the cell increasing. So you see this cell, the cell is going to become, cell is going to elongate. Then there's going to be cell elongation. And now we're coming to the last question, question 10a. Striated muscle is composed of myofibrils. Myofibrils contain several structural proteins, including troponin, tropomyosin, actin, and myosin. Outline the roles of these four structural proteins in the contraction of a sarcomere. Now, what will that be? Troponin binds to calcium and changes shape. Tropomyosin moves away from the binding site on actin. Myosin heads bind to form cross bridges with actin. Myosin head pulls the actin. Myosin head is an ATPase or binds to ATP to allow detachment. So troponin binds to calcium and changes shape. Tropomyosin moves away from binding sites on actin. Myosin head forms cross bridges with actin. Myosin head pulls actin. Myosin head binds to ATP to allow detachment. B part of the question, drugs that cause muscle paralysis, paralytic drugs are used during surgery to stop the patient moving. One commonly used paralytic drug is succinylcholine, which works by preventing contraction of muscles. Succinylcholine is able to prevent muscles contracting because it has a similar shape to acetylcholine. Suggest how succinylcholine is able to prevent muscles contracting. So basically what is going to happen, succinylcholine will bind to acetylcholine receptors. So sodium ion channels do not open, sodium ions do not enter the muscle cell, so sarcolemma is not depolarized. So the marking scheme is very clear. Succinylcholine binds to acetylcholine receptors. Sodium ion channels do not open, sodium ions do not enter muscle cells. Sarcolemma or the postsynaptic membrane is not depolarized. Part C of the question is Duchenne muscular dystrophy is a genetic disease that is caused by a single gene, DMD, affects striated muscles. The symptoms of the disease first appear at an early age. A fibrous protein, dystrophin, stabilizes muscle fiber during contraction. A person with DMD produces non-functioning dystrophin or no dystrophin at all. The disease occurs in about 4 in 100,000 people and mainly affects boys. Suggest and explain why boys are more likely to have DMD than girls. Well, I mean, the question is giving you some clues, and you've got to understand what are the clues. So it's a sex-linked gene which is on the X chromosome. It's a recessive allele. Males only have one copy of the gene, the X chromosome. Female are heterozygous, who do not have DMD, they are carriers. Only homozygous females will have DMD. So, single gene, and they've given you all that information. So the fact that they're saying boys are more likely to have it, so it has to be a sex linked or a gene must be on the X chromosome. It would be a recessive allele. We've done this in hemophilia. So sex linked gene on the X chromosome is a recessive allele. Men only have one copy because the Y chromosome there's no corresponding part. Female who are heterozygous will not have DMD, will be carriers, and the only homozygous if they both have the small recessive allele, the small d. Now, if we say, the small d causes it, so it will be X. Men would have to have this, and Y does not have anything. But a woman, if she has D and X big D, then she does not have the disease. But if then the woman, if she has this and she has, so she is a carrier. She's normal. She's a carrier. And the woman with this will be a sufferer. So sex-linked gene, X chromosome, recessive alleles, males only have one copy. Females who are heterozygous will be carriers. These women will be carriers. Only homozygous females will have small d. Small d will be, of course, will have the disease called DMD or Duchenne muscular dystrophy. So sex-linked gene on X chromosome, it's a recessive allele. Males only have one X chromosome. Female who are heterozygous do not have DMD. And this is, of course, will be the same marking scheme point, so it's not a separate marking scheme point. If you said females heterozygous do not have DMD, only homozygous females have DMD. So this would be, of course, this is a total of 10 marks, and that finishes this paper. And I hope these questions are a little easier for you now that you've understood how I've solved them. And thanks a lot, and wish you all the very best.